Okay, welcome everybody. This is chapter 10 on the nervous tissue in the brain. And what we're going to do is address the setup and the functions of the nervous system. Now, the nervous system is an extremely complex communication network, and it's usually the thickest chapter in any anatomy and physiology textbook because it has so many jobs to do. We'll talk about the components of the nervous system, where they're located, what they look like, and then we'll talk about how they work functionally. Okay, so the two anatomical divisions of the nervous system are the central and the peripheral nervous system. The central nervous system is made up of the brain, the brainstem, and the spinal cord, and the peripheral nervous system is made up of the peripheral nerves, which is everything else, and that connects the rest of the body to the central nervous system. The job of the nervous system is to take stimuli, which are physical changes in the environment or inside the body, okay, and turn them into electrical information, and then pipe that electrical information into the central nervous system, where it then ascends, goes up to the brainstem and the brain, and then a control center, which will either be the brain in the case of information that we're consciously aware of, or it'll be the brainstem and the hypothalamus in the case of information that we are not consciously aware of, will take those pieces of electrical information and combine them into an idea of what's happening to us. And then an appropriate command or commands will be generated, and that electrical information will be sent down the spinal cord to the peripheral nerves to the target, and then the target will do its thing. Okay? So, if we think about um, the world around us, right? Um, we are constantly bombarded with changes, right? We experience sound, sight, hearing, balance, touch, pressure, um, hot, cold, all these things. And then there's also changes that are going on inside our body all the time. Most of these changes would never make it to our conscious awareness. They're filtered out by a structure in the brain called the thalamus. And only the important stuff that we need to be consciously aware of and act on makes it to our conscious awareness. The other information is either filtered out and not dealt with, or is dealt with at the subconscious level by a functional division of the nervous system, which is called the autonomic nervous system. Okay, and so when you think autonomic, think automatic and subconscious, and that basically is sort of an autopilot version of controlling homeostasis in the body. It's able to function without us having to think about it, and thank goodness that's the case, because if we had to consciously control all our body's internal functions, that's all we would do all day. Okay, is control our heart rate and our blood pressure and our glandular secretions and everything else, and we wouldn't have the ability to really interact meaningfully with the world around us. Okay, so if we look at the cells that make up the nervous system, in the central nervous system, we have um, neuroglia and neurons. The neurons do the electrical work, and the neuroglia help the neurons do that work because they're so busy with processing and dealing with electrical information that they really don't have time to do a lot of the normal functions that cells carry out in order to um, manage to function every day. Okay, neurons do not regenerate. You get all you're going to get when you're born, and all that happens as you age is they begin to die. And so that's why they need all this extra help. In the central nervous system, the glial cells that help this out are called the astrocytes, the ependymal cells, the microglia. Okay, and the oligodendrocytes. Okay, what do they do? Well, the astrocytes essentially do the grocery shopping and the waste removal for the neurons in the central nervous system. So they provide nutrients, they remove waste products, they adjust the extracellular environment, okay? And they're also responsible for establishing something called the blood-brain barrier, where we keep harmful stuff that's dissolved in the plasma away from nervous tissue in order to protect it because it's lost the ability really to repair itself. The ependymal cells line hollow chambers inside the central nervous system, and they help create something called cerebrospinal fluid, and they also help to create the blood-brain barrier. Okay, so the cerebrospinal fluid is just a liquid in which the entire central nervous system floats, and it flushes out waste products, and it serves as a way for important stuff like oxygen and carbon dioxide to do diffusion. Okay, it has a nice pH and temperature and electrolyte concentration that lets the nerve cells do their thing. Okay, it's generated as a filtrate of blood, and then it is returned to the blood in special structures called arachnoid granulations, which are between the two big hemispheres of the brain. And we'll get to that when we talk about the brain in the next installment. The microglia are basically the only immune protection that the central nervous system has. And so they eat up dead cells and they try to attack and destroy pathogens. It's not a tremendously effective immune function for the central nervous system, which is why if the blood-brain barrier is broken and our immune system gets in there, the immune system for the rest of our body, it'll attack and destroy nervous tissue because it's never seen it before, and it assumes that it is harmful. Okay, this is why if you have a spinal cord injury, the immune system actually moves in and destroys neurons and starts to lay down scar tissue, and that may patch the wound, but that doesn't restore function. The oligodendrocytes are cells that form a sheath around the axons in order to insulate and protect it. Okay, oligodendrocytes create a substance called myelin. The myelin is an insulator for the axons, and that allows the current that flows through the axons to go much more rapidly from where it starts to where it stops. Okay, this is critically important in a phenomenon that we'll get to in a minute called an action potential. In the peripheral nervous system, the Schwann cells and the satellite cells are responsible for protecting the neurons. The satellite cells are around the cell bodies of the neurons in the peripheral nervous system, and they protect and nourish them. Okay, and the Schwann cells create the myelin sheath in the peripheral nervous system, which serves the same function as the oligodendrocyte myelin sheath in the central nervous system. Now, not all neurons are myelinated. Okay, some neurons in the central and the peripheral nervous system have little to no myelination, but they still have insulation and protection from connective tissue that surrounds and binds them together. Okay, in the peripheral nervous system, there's connective tissue layer around every nerve fiber called endoneurium. Bundles of nerve fibers are surrounded by perineurium, and the whole nerve is surrounded by epineurium. And the purpose of that is not only to kind of latch everything together, but also to provide a degree of insulation so that you don't have short circuits between information that's going in different directions in a single nerve. The same is true in the central nervous system. not all of the axons are myelinated, but they all are surrounded by a protective sheath. Oligodendrocytes can wrap around um, axons in the central nervous system, but not necessarily produce lots of myelin. Okay, but still insulate and protect the axons from each other. Okay? If we look at the different parts of a neuron, this is a typical multipolar neuron because it has multiple extensions coming off of its cell body. We see short extensions that are called dendrites, which are the receiving end of electrical signals, and then this long extension called an axon, which is the sending part. Okay, so the way that this works is that there are hundreds of other neurons surrounding these cells, and they're all talking to it through um, little gaps between the axon terminals of one neuron and the dendrites and somas of another neuron, and this is a special little gap called a synapse. Okay, and what happens when we generate a nerve signal is that a chemical called a neurotransmitter is sprayed onto the dendrites or the soma, and that creates a current that, if it's sufficiently strong, will ripple across the membrane of the soma to this little area here, which is called the hillock or the trigger zone. And if we reach a specific voltage, we generate a long-distance electrical signal called an action potential that goes from the hillock all the way to the axon terminal, and that results in neurotransmitter release onto the target. Okay, so this is the receiving part, this is the sending part, and you could think of all the rest of this as kind of the integrating part. Okay, a nerve cell can be really long. Okay, it can be a foot long or longer, and so it has to have a special setup to move stuff from the soma to the terminal, and from the terminal back, and those are essentially cytoskeletal components that are transport proteins. They're like little train tracks to move stuff back and forth. Okay. The axon is the sending part of the neuron, and if it is myelinated, it's going to be a high-speed electrical signal. Okay, the action potential, which is the long-distance signal we talked about, can move at top speeds of 250 miles an hour in the fastest neurons. Okay, and those are the neurons that do things like send information to our skeletal muscles so we can move, and bring information from our joints and our muscles into the central nervous system so that we are able to do things like maintain our balance. Okay, in a myelinated axon, there are little spaces that are not insulated, and those are called nodes of Ravnier. Okay, and those are areas where action potentials are formed in a process that's called saltatory conduction, which we'll get into in a bit. Okay, the axon terminal is where little swellings called synaptic boutons or telodendria are located, and they release neurotransmitter when the action potential reaches them and spray it onto the target. Okay, the neurolemma is simply the outer sheath of Schwann cells that forms this little tube, okay, the myelin sheath, which can help in peripheral nerves to regenerate the axon if it's clipped or damaged. Okay. Different kinds of neurons do different jobs. Sensory or afferent neurons carry information from the body into the central nervous system. Efferent neurons carry motor commands out to the targets so that they carry out our instructions. And interneurons, which are in the central nervous system, process electrical information as it's either heading up to the brain and brainstem or coming down from the brain and the brainstem. okay? And that's found primarily in what's called the gray matter of the central nervous system, which is the inner neurons that are unmyelinated, the somas, the dendrites, and the white matter in the central nervous system are the myelinated axons. So I always tell my students that in the central nervous system, the gray matter is for signal processing and the white matter is for signal transmission. Okay? All right. If we talk about motor neurons for a minute, the cell bodies of the motor neurons are found in the central nervous system. Okay, so anything that has outgoing information is going to have its cell body in the gray matter of the spinal cord, which is in the core of the spinal cord. The sensory neurons have their cell bodies in the peripheral nervous system. Okay, an example would be the dorsal root ganglia, which are collections of nerve cell bodies that are on the back side of the spinal cord outside the vertebral column. Okay, the autonomic nervous system, which is the nervous system that operates without our conscious input, has its nerve cell bodies in what are called the lateral horns of the gray matter of the spinal cord, and then a two neuron chain carries that information to the target, which is anything but skeletal muscle. In the sympathetic division, in the parasympathetic division, little bits of gray matter called nuclei, which are embedded in white matter up near the brainstem or down near the sacral spinal cord, carry information in a two neuron chain out to the target. Okay. So that's the skinny on white and gray matter, right? White matter for signal transmission in the central nervous system, gray matter for signal processing. In the peripheral nervous system, the neurons are basically sending axons out to their targets in the efferent division, or bringing axons in from the body in the afferent division. Okay, so when you look at a nerve, what you're looking at are bundles of axons, and depending on whether we're looking at a nerve that carries autonomic information or somatic motor information or what have you, it could be any combination of incoming and outgoing information, and it could be any combination of somatic or autonomic information. And so you can have bundles of myelinated axons, you can have bundles of myelinated and unmyelinated axons, all within a single nerve. Okay, most nerves in the body are mixed nerves, meaning they carry both incoming and outgoing information, and they can carry any combination of somatic motor or autonomic information. It all depends on what nerve we're talking about. Okay, and then there are some nerves that are strictly efferent and some that are strictly afferent. Okay, efferent nerves only carry motor commands out. Afferent nerves only carry sensory information in, and most of those nerves are part of a group known as the cranial nerves, and we'll learn about them a little bit later as well. Okay, all right, the next three installments are going to talk about how it is that a neuron generates an electrical signal and then sends it to a target. Okay, so this talks about membrane potential, and then action potentials, and then the results of all of those processes. So listen up.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body, in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts. Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, Action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
Okay. Now we're going to talk a little bit about the brain. Okay. The brain is an integration center. Okay. It's made up of the cerebrum, the diencephalon, and then underneath the brain we have the brain stem, and then we have a structure called the cerebellum. Okay, and you can see those areas here. Right. The cerebral cortex is divided into lobes. The frontal lobe is under the frontal bone, the parietal lobe under the parietal bone, the occipital lobe under the occipital bone, the temporal lobe under the temporal bone, and there's a fifth lobe called the insula, which is buried in this little indentation between the temporal and the parietal lobe. Okay. The cerebellum back here looks like a little hunk of turkey burger underneath the occipital lobe. The brainstem here in green, okay, and the diencephalon here in purple. Okay. The parts of the diencephalon are the thalamus, the hypothalamus, the epithalamus, and the subthalamus. Okay. The parts of the brainstem are the midbrain, the pons, and the medulla oblongata, and then there's the cerebellum back here. The reason that the brain surface is wrinkly is to increase the surface area so that it can do more electrical work in a tinier volume. Okay? The main source for energy in the brain is glucose. Okay? So sugar is brain food. And so when the body is kind of stressed for nutrients, one of the things that it'll do is it'll always save sugar for the nervous system, and then tissues that can use something else will use something else. Okay? The brain is gray matter on the outside and white matter in the core. Okay. Remember that the gray matter is for signal processing and the white matter is for signal transmission. The same arrangement is in the cerebellum. Gray matter on the outside and the white matter in the core is called the arbor vitae. Okay. Remember that when we're talking about the cerebral cortex, we're talking about higher brain function. So this is where we perform tasks like learning, reasoning, language, and memory. This is where we become consciously aware of stimuli that happen inside us and on our body surface and outside in the environment. Okay. And we integrate all that stuff and then we send it to the part of the brain where we decide what we want to do about it, which is the frontal lobe, and then we generate a motor action plan, and then we send that into two places, the cerebellum and the spinal cord, and then the peripheral nerves carry that information out to the muscles, and we do our thing. Okay, so that's for stimuli that we're consciously aware of, right? For stimuli that are not consciously acknowledged, okay, but are important nonetheless, the hypothalamus and the brainstem take care of that information and send the commands to the targets through a division called the autonomic nervous system. Okay. The cerebrum has gyri and sulci. Okay. The the gyri are the ridges in the cerebrum, and the sulci are the shallow indentations, and then the fissures are the deep indentations. Okay. Um, there are several fissures that are important. There is a longitudinal fissure that divides the brain into two hemispheres. There is a central sulcus that divides the frontal from the parietal lobe, okay? And then there is a lateral sulcus that divides the temporal lobe from the parietal and the frontal. And then there is a transverse fissure that divides the cerebrum from the occipital and the temporal lobe. Okay, and again, the reason for all of these convolutions and ridges is to increase the surface area of the brain. All right, so there's the different lobes. Okay, um, the transverse fissure would be here. There's the lateral sulcus, the central sulcus, right? And then the longitudinal fissure is only visible from the top, and it would run right across here. Okay? Um, Different lobes of the cerebrum do different jobs. Okay, the frontal lobe is where our will, our reasoning, our planning, and our ability to produce motor commands to skeletal muscle are housed. Okay, our intellect is in here. Um, our our better self is in here. Our super ego is in here. Okay, if you've ever taken a psychology course, okay, our judgment is in here. Um, the parietal lobe is an area of the brain where we take stimuli that come from the surface of the body and our muscles and our joints, and um, we we apply meaning to it, okay, and then we integrate it with information from the other lobes, like the temporal, the occipital, the insula, and so on. And we get an idea of what's happening to us, and we send that information to the frontal lobe, where we determine what we want to do about it. Okay. The occipital lobe is for the processing of visual information, and the temporal lobe is responsible for our ability to hear, some of our ability to maintain our balance along with the parietal lobe, our ability to smell, okay? Um, and the insula, which is a fifth lobe kind of buried under this crevice, under the lateral sulcus, is responsible for our sense of taste and our visceral sensation, which is coming from our internal organs, okay? So there's the frontal lobe, right? What it does and why it does it, okay? Now you might wonder, how do we know all this stuff? Well, most of the information that we know about the frontal lobe comes from brain damage and animal models, okay? So we kind of have to take that information with a grain of salt, because the brain and the brainstem and the spinal cord are one of the few organs in the body that we can't take out, work on, and put back and see what happens. Okay, they're sort of locked in position, and we can't do anything with them. So when you damage a piece of this system, and then you lose certain functions, um, it may or may not be an oversimplification to say that those functions were in the damaged area. Okay, it might be that those functions simply passed through the damaged area. So a lot of this, again, there's a little bit of gray area and guesswork involved in it. Some of it we know for sure. Some of it is a little fuzzy. Okay, So in case you were wondering about that. Motor activity takes place in the frontal lobe. Um, the, the, the motor association area is, is right behind the very tip of the frontal lobe, and that's where we form motor action plans. And then the motor cortex is in the precentral gyrus right in front of the central sulcus, and that's where we send the motor action plans before they're going to head down to the cerebellum and the spinal cord. Okay. The motor speech area is usually on the left side of the frontal lobe, and it's called Broca's area. Okay. And then the frontal eye fields are also located there. Okay. So you can see again the different areas of the cerebrum and what they do. Okay. There's Broca's, there's your frontal eye field, right? There's your pre and post central gyrus, right? This is the somatosensory association area. This is the somatosensory cortex, okay? Our taste area is, again, going to be in the insula. There's a little bit of it in the parietal lobe as well, okay? There's your visual and your visual association. There's your auditory and auditory association, your olfactory area, okay? In the frontal lobe, in the precentral gyrus, we have regions of the precentral gyrus that are dedicated to the movement of different body parts. And the body parts are not proportional to our body proportions because certain parts of our body need more motor control than other parts. And so the face, the hands, the lips, the tongue are overrepresented, and the rest of the body is underrepresented. And so that forms something called the motor homunculus, right, which is in the lower right here, representing the body proportions for motor control. And you can compare that to our normal body proportions and see where the greatest degree of motor control exists. Okay, and that shouldn't surprise you that we've got a lot in our hands and in our lips and in our face because we do a lot of complex movements with those parts of our body. Okay. The other reason, of course, that there's so much brain area dedicated to that is that there's more neurons bringing information in from those regions, and so you need more brain surface area to deal with it. Okay. How do we know this? Well, we know this through direct experimentation by stimulating those parts of the brain and seeing actual motor responses. So this is one of the few aspects of the brain that we're pretty solid on. Okay. The parietal lobe we talked about is somatosensory and also has some gustatory function. Okay, and it's also a multimodal association area. The temporal is where our auditory and our olfactory is. The gustatory area, again, for the sake of our textbook, we'll say it's in the temporal lobe, but most textbooks assign it to the insula. And of course, the occipital lobe is where our vision is processed. Okay. Speech areas involve the frontal, the parietal, and the occipital lobes. Okay, they're often in the left hemisphere. And then there are association areas that allow us to interpret sensory information. Examples include visual, auditory, and somatosensory association areas. Okay, in most individuals, okay, the skill sets are on different sides of the brain. Okay, different sides of the cerebrum. The left brain and the right brain deal with slightly different things. The right brain deals with spatial localization, artistic ability, musical ability, and so on. The left side of the brain is primarily for logic, reasoning, and computation. Okay, but that's not in everybody. All right, that's just in most people. Okay? The other important point about the way that the cerebrum is set up is that the left side of the cerebrum controls the right side of the body and vice versa. And the same is true for sensory information. Sensory information from the right side of the body goes to the left hemisphere and vice versa. And the reasons for that are not entirely clear, okay? It just is the way we're set up. Um, that said, the brain does have communication between the two hemispheres through tracts of white matter, the corpus callosum, and the fornix are examples of that, okay? So as soon as something happens in one hemisphere, information about that is relayed to the other hemisphere, and that allows us to perform coordinated movement and integrate stuff that's going on in the brain. If we damage those connections, then the two halves of the brain can start to operate independently of each other, and you'll get two sides of the body operating as if they don't know each other. Okay, and that's an example of a condition called phantom hand, where you'll have your right hand doing something that your left hand doesn't realize, and it may completely oppose what the other hand is doing. In some cases, when that happens, people stop acknowledging one side of their body as belonging to them, and they treat it like a shadow or a reflection. Okay, and that seems odd to us because we have an integrated brain, but this is what happens when the damage occurs. Okay, there's two parts of the diencephalon: the thalamus, and then the other structures, which are the hypothalamus, the epithalamus, the subthalamus. The thalamus is a sensory pathway for the information coming from the lower brain and the spinal cord to the sensory areas of the cerebrum, and it's critically important regarding pain sensation as well. The hypothalamus sits above the pituitary gland and below the thalamus and controls the endocrine system, and it's also the governor of the autonomic nervous system. Okay, so this is, in the hypothalamus, sort of almost a second brain. Okay, this is the brain that works on keeping our homeostasis operating. Okay, our blood pressure, our heart rate, and so on. Okay, the the um, the epithalamus lies above the thalamus, and the subthalamus lies just behind and below the thalamus, okay, and sort of above the hypothalamus. The brain stem has three major parts. The midbrain connects the diencephalon to the brainstem. The pons is below the midbrain, and the medulla oblongata is below the pons. Okay. The midbrain has clusters of gray matter that serve as reflex centers for vision and hearing. Okay, and these are called the colliculi. The superior colliculi deal with visual reflexes, and the inferior colliculi deal with auditory reflexes, and they are on the back of the midbrain. The pons is critical for regulating breathing rate, and it's also a connection between the cerebrum, the brainstem, the cerebellum, and the medulla oblongata. So there's a lot of white matter in the pons. Okay, and the medulla oblongata, which is the lowest part of the brainstem, contains nuclei that control heart rate, blood pressure, respiration, vomiting. Okay, and it also has a critical role in interacting with the thalamus and even the pons in the midbrain to control sleep and wakefulness. Okay, a column of gray matter called the reticular activating system runs through the back of the brainstem from the medulla oblongata all the way up to the midbrain, and it works with the thalamus to control when we wake up and go to sleep. Okay, so the way this works is that as light levels go down, the eyes pick that up, and they communicate via the hypothalamus to a gland called the pineal gland, which is on the back of the brainstem. And the pineal gland produces a hormone called melatonin, which levels go up when it gets dark, and that helps the reticular activating system to tell the thalamus to close the gates of perception to the cerebral cortex, and we become less aware of what's going on around us and we fall asleep. Okay? And then when light levels come up, melatonin levels drop, and then the reticular activating system can tell the thalamus to allow stimuli to get to our cerebral cortex, and we wake up. Okay? So that's an important other function. Okay? It's also involved in pain transmission, right? It helps to reduce the perception of pain through neurotransmitters that are called enkephalins and endorphins. Okay? The medulla is also where motor commands cross from left to right and right to left, okay, as they head down the spinal cord, and where sensory information crosses on the back of the medulla as it's heading up to the brain, to the thalamus and into the brain. Okay. The cerebellum is connected to the brainstem by 3 peduncles, which are connections that allow the cerebellum to receive, integrate, and deliver information to different parts of the brain and the spinal cord. The primary responsibility of the cerebellum is coordinating muscle activity to produce coordinated muscle responses after integrating incoming information from your eyes, your balance apparatus, your muscles, and your joints. Okay? Essentially what it does is it compares the motor action plan that's received by the precentral gyrus to what you're actually doing. And if the two don't match up, it fixes that plan and sends it back to the frontal lobe. And then we repeat the process of sending it down to the brainstem and the spinal cord. Okay? And that way we move in a smooth and coordinated fashion. If the cerebellum is damaged, you end up with a condition called cerebellar ataxia, where it's very difficult for you to initiate and stop voluntary movement, and there's a lot of unintended movement in your actions. Okay? It's also an important reflex center. The limbic system is our emotional brain. It's a wishbone-shaped group of structures formed by parts of the cerebrum and the diencephalon. So it includes the cingulate and dentate gyrus, the hippocampus, the olfactory bulb and tract, and the amygdala. Okay? And what it does is it attaches emotional content to our experience. So, for instance, if you smell perfume and you have a functioning limbic system, then you would say that smells nice. Okay? And the nice is a judgment call. Whereas if you didn't have a functioning limbic system, you would smell the perfume and you would say that smells like perfume. A condition in which the limbic system does not work properly is autism. Okay? In autism, individuals have a difficult time expressing their emotions or interpreting emotions in the actions and facial expressions of others. The reticular formation is responsible for maintaining consciousness and activating the sleep-wake cycle. It also has our gaze center in it. This is what's working with the thalamus to allow us to have consciousness or to fall asleep. And again, the melatonin levels are a big part of that as well, which means the hypothalamus and the eyes have a role in this. Okay? Note that consciousness is wakefulness and is dependent on the reticular activating system. We can, if we damage the reticular activating system, fall into a coma. Okay? And then there are memory areas in the limbic system, the hippocampus, for instance, is like the body's DVR. And what it does is it stores information that you've gathered during the day, and then as you're sleeping, it plays it back into appropriate areas of the brain that store memory. Those would be the association areas. And that's one of the reasons why our dreams often are composed of disconnected events that we experienced that day. All right, you could think of that as seeing the information going from the hippocampus to the appropriate area of the brain. Okay, so that's one of the reasons, a lot of times, why a lot of our dreams don't make sense to us. Okay? Sleep is a restorative mechanism. It's thought that it helps to regenerate neurotransmitter levels in the brain. The different stages of sleep include non-rapid eye movement sleep and rapid eye movement sleep. Okay? The 4 stages of non-rapid eye movement sleep progress from light to deep. REM sleep is characterized by changing blood pressure, changing respiratory rates, rhythm, and pulse rates. The obvious characteristic is the rapid eye movement, and you could think of that as you looking around in your dreams, okay? And it's also the most restorative part of sleep. So the quicker you get to REM sleep, the more rested you're going to feel. This is one of the reasons why, if you're a predator, okay, like a dog or a cat, you can fall asleep and go into REM sleep at the drop of a hat, and that will be restorative enough for you to be able to immediately wake up when you hear something or feel something and respond as if it were a threat or as if it were something that you want to attack, kill, and eat. Okay? Memory areas in the brain are located in different physical locations. Immediate memory leads to short-term and then long-term memory. Recollection of thoughts and images are going to progress through these different regions of the brain. When they get to long-term memory, what will happen is that you'll be able to draw on them in the future, okay, in order to apply meaning to the stimuli that you experience. Okay? We know that they're in different physical locations of the brain because different conditions that damage the brain alter different memory areas. An example would be amnesia, where you have no long-term memory but your short-term memory is unaffected, and dementia, in which your short-term memory is virtually non-existent but your long-term memory stays intact, and those affect different parts of the brain. There are layers of protection for the central nervous system that provide the ability to avoid damage via trauma. We have bone, of course, surrounding the entire central nervous system. The brain and the brainstem are contained in the cranium, and then the meninges are 3 sets of membranes that surround the entire central nervous system and stabilize it and serve as a way for blood vessels to provide nourishment and oxygen to the central nervous system structures, even though the blood they contain never comes into contact with the nervous tissue. And it also helps to tether the central nervous system structures in place so that they don't damage themselves by bumping against the hard surfaces. And then the cerebrospinal fluid is a filtrate of blood in which all of these structures float, and it provides an environment in which they like to function and serves as a way to bring in diffusible components like oxygen and bring out things like CO2, and also has a nice temperature and pH and electrolyte concentration that allows the nervous tissue to function properly. Okay, and then we've already discussed the blood-brain barrier. What you're looking at here is just a diagram of the membranes that protect the central nervous system, and they are from superficial to deep: the dura mater, the arachnoid mater, and the pia mater. In the brain, the dura mater is a double-layered fibrous membrane that surrounds the outer covering of the brain and the brainstem, and has between the hemispheres a blood-filled space called the dural venous sinus, and it attaches to the floor of the cranium to the ethmoid bone to a ridge called the crista galli, and that sort of tethers the brain in place while it floats in CSF so that it doesn't damage itself. Beneath it is the arachnoid mater. It has spider web-like extensions between the pia and the surface, the inner surface of the arachnoid. The subarachnoid space that surrounds the entire central nervous system is filled with cerebrospinal fluid, again providing cushioning, an appropriate temperature, pH, and electrolyte concentration, and a diffusible medium for CO2 and oxygen. And so the neurons work best in an environment like that. And remember that it also keeps the central nervous system tissue isolated from the blood supply. Okay, so the only material that goes from the blood into the neurons, other than nonpolar substances, are what the astrocytes and the ependymal cells dictate. The dura mater is thick, tough connective tissue, and it divides into sinuses that are filled with blood. The subdural space is located beneath the dura mater, and it does contain blood. A clinical application for this is that when a person sustains a sharp blow to the head and develops a subdural hematoma, one of the dangers, of course, can be increased pressure on the brain. Okay, since the cranium can't expand, this could lead to coma and death, right? If it's not quickly treated. Normally, when we see an increase in intracranial pressure, one of the ways we deal with it is to literally drill holes in the cranium in order to let the extra pressure out. Okay. The cerebrospinal fluid is formed in each of the ventricles that are contained in the core of the brain. They include the two lateral ventricles, and then between them is the third ventricle connected by the interventricular foramina. And then the third ventricle has a cerebral aqueduct which leads to the fourth ventricle, which is behind the pons and in front of the cerebellum. And that fourth ventricle is continuous with the central canal of the spinal cord, which projects all the way down through the middle of the gray matter. And it also has median and lateral apertures that let the cerebrospinal fluid escape the fourth ventricle and bathe the entire outside of the central nervous system, flowing through the subarachnoid space. And this fluid rejoins the blood supply in the dural venous sinus, which is a blood-filled area between the two layers of dura surrounding the brain, where little protrusions called arachnoid granulations let that fluid re-enter the blood supply so that we have a continuous flow. And so that has a flushing effect. The CSF is made of water, glucose, protein, and sodium and chloride ions. We'll look at a diagram that shows the ventricles and the flow in just a minute. The ventricles of the brain are lined with ependymal cells. You can see them here, okay? The two lateral ventricles, this is the interventricular foramina, this is the third ventricle, and then this is the cerebral aqueduct. Right there is the fourth ventricle, okay? And this is the central canal that's continuous with the fourth ventricle, okay? Basically, we create CSF in each of these chambers at structures that are called choroplexuses, okay? And then they flow from lateral to third, from third to aqueduct, from aqueduct to fourth, from fourth to central canal, and then out the median and lateral apertures to bathe the entire central nervous system, okay? And you can see the flow here, all right? See how the CSF is secreted by the choroid plexus at 1, flows through the interventricular foramina into the third ventricle at 2, okay? The choroid plexus in the third ventricle adds more cerebrospinal fluid at 3, flows down the aqueduct to the fourth ventricle at 4, and the fourth ventricle adds more CSF, and then it flows out those apertures and bathes the outside of the central nervous system in the subarachnoid space. And then the arachnoid villi, which are shown up there by 8, return the cerebrospinal fluid to the blood supply. Okay, and so it's a constant flow. If that flow is impeded, then the cranial pressure builds up, and that can be an immediate medical emergency. Okay, in newborns, since the flat bones of the skull have yet to fuse, what happens is that the cranial vault expands, so it looks a little bit like a light bulb, and that's a condition called water on the brain. And to fix that, we put a shunt upstream of wherever the blockage of cerebrospinal fluid is located, and then we have a tube that drains into the abdominal pelvic cavity, and then the blood vessels of the abdominal pelvic cavity pick that fluid up, and then we are able to reduce the intracranial pressure, and the cranial vault normally goes back to its original dimensions at that point. Okay. The astrocytes and the ependymal cells help to isolate the central nervous system from toxins that are polar or charged that could get into the central nervous system, and so that helps to ensure that the brain, since it has minimal immune function, ends up safe. Okay. Often antibiotics have a difficult time crossing the blood-brain barrier, so an infection can be treated by selecting an antibiotic that is nonpolar, or by injecting the antibiotic into the subarachnoid space. In the event that you do get an infection in the central nervous system, it's very serious. Examples of potential complications include meningitis and encephalitis. And usually the way that this works is that either trauma breaches the blood-brain barrier, or there are certain organisms that can penetrate vulnerable spots in the blood-brain barrier and gain access to the central nervous system. And in order to diagnose, we often perform a spinal tap, where a relatively large gauge needle is inserted into the subarachnoid space, and we withdraw some of the cerebrospinal fluid that surrounds the spinal cord, and we determine whether or not there are pathogens in there through microscopic examination. And if there are, then we have to take immediate measures. Okay. Please let me know if you have any questions. I can respond by email within a day, and I will join everybody in the next podcast. I thank you for listening.